let you guys get on to what you need to get done. Okay, so let's start off talking about, all right, chapter 12. And we're going to talk more or less about the anatomy component of the neuron here. All right. So one of the things that I want you folks to keep in mind, all right, bless you, is we're going to see in the upcoming chapters, and we've seen it a little bit already, that there's, I call it the rule of threes. A lot of things that we're going to be discussing, like layers of muscle or layers to a nerve, are going to be in rules of three. Okay. So to understand, first of all, what the heck a nerve is, all right, we need to describe it. Okay. Because we're going to go into the specific parts. So when you hear me say axons, all right, that's going to be the once we do the labeling for the neuron, you'll understand what that is. All right, but you need to understand the definition of what a nerve is, because depending where we are, okay, in the nervous system, okay, there's two major structural components of the nervous system. You've got your central nervous system, that's your brain and your spinal cord, and then you got everything else, and that's called the peripheral nervous system, okay? So when we're talking about the peripheral nervous system, all right, when we're dealing with a structure that has a bundle, that's many, okay, a lot of parallel axons, we refer to that as a nerve, okay? You need to understand that because it's called something completely different in the central nervous system. It's called a tract, all right? So in the central nervous system, we would call that same thing a tract, okay? But we'll get there. We'll talk about that, all right? So in the peripheral nervous system, a bundle of axons, all right, is referred to as a nerve. And these nerves have three coverings to them, okay? So the outermost, epi is going to be the outermost. This is going to cover the entire nerve. So when you're dissecting a cadaver, all right, or if you're a surgeon and you're going in and you're moving structures around and you see a structure that is kind of white looking, okay, and it doesn't have a lumen, most likely that's a nerve. And so what you're looking at is the epineurium, right? The epineurium, okay, is going to be made up of that dense irregular connective tissue. So again, dense regular connective tissue, right? When we look at that, that has kind of a structural component to it. It adds somewhat of a protective type of covering to the nerve, but it also kind of gives that nerve a little bit of structure, all right? So it's just not, you know, flip-flopping all over the place, okay? The second layer, all right, we go just deep to the epineurium, all right? That's the perineurium. This is the middle layer. It's also made up of dense irregular connective tissue, okay? So now what we've done is we've taken just a few, all right, of those axons, all right, and we put them into a bundle, and we've bundled that together, and we call that a fascicle, okay? And then the most internal or deepest layer, all right, is the endoneurium. This is made up of a loose connective tissue, all right, areolar connective tissue. We're going to see a very similar, when we're talking about the coverings to muscles, we're going to see a very similar pattern with muscles, all right? You're going to see three layers of connective tissue. The innermost layer is going to be loose connective tissue, and then the middle and outer layer are going to be both dense irregular connective tissue. So whatever you're learning here for the neuron, you're going to learn for the muscles too, all right? But this endoneurium covers each individual axon, okay? And this is important, all right, because one, we have to separate these axons from one another specifically for this property, all right, that it electrically insulates. Because technically when we get into the neurons, you're going to learn that a nerve signal is an electrical impulse that's traveling down the axon, okay? And we'll get into the specifics about that. So we need to insulate axons from one another so we don't start discharging axons that we didn't mean to discharge. Okay, so if we insulate them from each other, then we're good. Our signal goes to where we want it, and we're happy. Okay. In addition to having connective tissue coverings, all right, our nerves need to be vascularized. Makes sense. They need nutrients just like anything else, any other type of soft tissue. All right. So we'll see these small blood vessels between, all right, the outer layer here, the epineurium, all right, and we'll also see it in the middle layer, the perineurium. Okay. So we'll see these blood capillaries so we can get our nutrients and get rid of our wastes, all right, for these axons and these neurons here. All right, so here's a quick picture. All right, so we'll start with the smallest structure first. This little individual rod that's kind of sliding out. That's the axon. That's one single nerve cell. Okay? So this is the axon, which is that long process that comes off the cell body. It's covered by the endoneurium. That's a loose connective tissue. All right, areolar connective tissue helps to insulate this nerve, keeps its electrical impulse to itself. All right, now what we've done is, when we bundle several of the axons together, all right, now we're going to be dealing with a fascicle. So that's what we're seeing up here. Here's a fascicle. This whole structure is a fascicle. So we have another layer of connective tissue goes around it. That's the perineurium. And then finally, when we're talking about the entire nerve itself, okay, that's going to have another layer. So we take all the fascicles, we bundle them together, and then we throw this epineurium around it. That's another dense irregular connective tissue there. Okay? So that's what we're seeing. If you want to look at it from this point of view too, all right, here's your spinal cord, okay? And then the nerves that come off the spinal cord, we call them spinal nerves, okay? And before a spinal nerve is actually made, you've got nerve fibers coming off of the back part of the spinal cord and nerve fibers that are coming off of the front. Then when they fuse together right here, all right, that is referred to as a spinal nerve. And a spinal nerve is what we call a mixed nerve. It means it has motor and sensory axons. So it carries information going to the central nervous system, and it also carries information leaving the central nervous system. All right. For the most part, sensory information comes to the central nervous system, and motor information leaves the central nervous system. Okay. Question?
Yeah. So if the nerve is an organ, is the axon the cell, the fascicle, the tissue, and then the nerve is the organ?
No, the nerve actually won't really be considered the organ. The brain will be considered an organ. You're asking me?
It says in the book that it's an organ.
The actual nerve itself?
Yeah.
Okay. How I teach is mainly as a cell, so it's really tough to read that sentence. What's it saying?
It says a nerve is an organ composed of cable-like bundles of axons, connective tissue layers, and blood vessels, and it's a component of the brain.
You said nerve or neuron?
Nerve.
Okay, okay. So, all right, let me rephrase my, yes. Okay. Neuron is the actual nervous tissue cell, okay? The nerve itself is made up of thousands of those actual cells that, perform a certain function. So your question was,
Like, would you consider the axon, the cell, the fascicle, the tissue, and the nerve the organ? Like, if we're trying to organize it?
oh, I see what you're saying. Okay, okay. So I would consider the whole neuron, that includes all the coverings, all right, all the insulation, even the blood vessels that are found inside there, that's going to be your actual organ tissue itself, all right? And then when you break it down to the cellular tissue, because in a neuron, you're going to have not just, in a nerve, you're not just going to have neurons, you'll also have glial cells, which we're going to talk about here in a second. Those are also cells in the central nervous system there, and the peripheral nervous system. All right, let's jump into nerves and ganglia. Okay, so kind of a breakdown, so we give you a little bit more of a general idea when we're talking about nerves and ganglia. Okay, these two terms, all right, are pretty much referring to the peripheral nervous system. All right, so a nerve is, like I said, it's a bundle of axons. A ganglia, okay, is going to be, all right, where one neuron synapses, that means where it connects to another neuron, okay? So in the peripheral nervous system, we call that a ganglia. In the central nervous system, we call that a nucleus. So not to make things more complicated, all right? But a nucleus is to the central nervous system as a ganglia is to the peripheral nervous system. All right, but we'll talk more about that. All right, so when we're talking about the structural classification of nerves, that's going to be the anatomical function, okay? So that means, real simple, if you see a nerve coming off the brain, we refer to that as a cranial nerve, okay? Keep it simple. If you see it coming off of the spinal cord, then it's a spinal nerve, okay? Both will be, well, I shouldn't say both, all right? Spinal nerves are mixed nerves. Not all the cranial nerves are mixed nerves. You have some cranial nerves which are motor nerves, some are sensory, then some are both. All right, but to keep it real easy, if you see a nerve coming off the brain, it's a cranial nerve. All right, if you see it coming off the spinal cord, it's a spinal nerve. All right, so when we get down into the functionality of these nerves, all right, a nerve can either be sensory, okay, that means it's going to be bringing information to the central nervous system. It can be motor. All right, that means it's going to take information from the central nervous system and send it out to an effector organ, or it can be both. It has sensory and motor components there, okay? So when you hear this term down here, all right, this bullet says most named nerves are in this category. Sciatic nerve is a named nerve. That's a mixed nerve, all right? Your median nerve, that's the one that gives you carpal tunnel. That's a mixed nerve. All right, musculocutaneous, that's the nerve that innervates the biceps. I could go on and on. All right, so when you hear a named nerve, all right, that pretty much is going to be a mixed nerve, okay? All right, so again, when we refer to this term, all right, a ganglion is a cluster of neuron cell bodies. That's usually where you're going to see a synapsing. All right, the cell bodies are where you're going to see, all right, a synapsing where one neuron connects to another. All right, so you get a clustering of that. All right, we call that a ganglion, and that's in the peripheral nervous system. When we get a cluster of the cell bodies in the central nervous system, then we call that a nucleus. Okay? All right, characteristics of neurons. And this is very important that you understand this because some of this is going to apply to muscles too. All right? So we have excitability, conductivity, secretion, extreme longevity, and amitotic, all right? So excitability is basically your nerve, when it's at rest and it's not doing anything, all right, minding its own business, it's in what we call the resting state. Nothing is happening, all right? Something needs to stimulate that nerve, all right? So going back to chapter 1, okay, you need a stimulus of some sort. It could be, depending on whatever the if it's a sensory neuron or a sensory nerve, it could be a pain stimuli. All right. If it's movement related, it could be a mechanoreceptor. All right. Point being is we need a stimulus. All right. And then that stimulus is now going to affect some changes in the neuron, and we'll talk about those changes. It's going to change what we call the resting membrane potential, which is the charge across the plasma membrane. Okay. It's a specific charge. We'll get into that. So this is what we're saying. We're going to change that cell's membrane potential. Okay. It's going to go from a certain number, all right, which is actually negative 70 millivolts, and it could change it to plus 30, let's just say, all right? So we need that stimulus to excite the nerve cell, and then we get a nerve signal going on, all right? So a neuron, just think of it as like a wire in your house, all right, like your internet wire, or if you have an Ethernet, okay? So your cable signal has to come into your house. All right, it gets conducted along a coax cable, most likely, and enters into the modem. Same thing here. All right, your nerve needs to be able to conduct its signal along its entire pathway there. Okay, so in order to do that, okay, it has to send that information from one region to another region to another region to another region, and it has to sustain that, or it needs to propagate that electrical signal. Okay, so again, I know this is stimulating you guys because it's going to be really exciting when we learn about this, but we're going to learn about these voltage gated channels. We learned about those in chapter 4. Remember those membrane, those integral proteins in the plasma membrane there, and how something will stimulate those channels to open up, and then that channel will let something enter or leave the cell. Well, that's what we're going to get into here, all right, later on. Okay, secretion, this is where we see at the end of the nerve, down by the nerve, the part of the nerve in what we call the axon terminal, all right? And we have this neurotransmitter, which is a chemical, and it's stored there in these little bubbles, all right? And then when the nerve signal gets down to that end, it's going to stimulate those bubbles to release that neurotransmitter, all right, out of the cell, and it's going to release it onto either another neuron to stimulate that neuron or onto an effector organ. It could be a muscle, it could be a gland, all right, some sort of cell to do whatever it's supposed to do, all right? Extreme longevity, pretty much most of the cells, the neurons that you're born with, you keep all throughout your life, all right? They do not divide, all right, past that point where you are actually done, I don't want to say done growing, but pretty much when you're developing and you've been born, those neurons that you have are the ones that are going to carry with you for the rest of your life. Neurons are considered to be amitotic. They're not considered to divide. All right? Certain cells will divide, all right? But neurons do not, except there's always exceptions to the rule, and we'll talk about that in extreme cases. But there are some neurons in certain parts of your central nervous system that actually do divide, okay? All right, so let's talk about the different parts of the neuron, okay? The cell body, also known as the soma, okay? This is the part that is actually going to have, all right, the nucleus, our control center there, the endoplasmic reticulum, all right? We're also going to see cytoplasm. We give it a special name. You need to know this name, all right? Perikaryon, okay? You're going to find when we go into the neurons and into muscle, we have to name the cytoplasm something different, all right, in these cells, okay? In the muscle cell, we call it sarcoplasm. Here in the neuron, all right, in the cell body, we call it perikaryon, okay? So the cell body, all right, is the receptive segment of a neuron, which means it's going to get information somehow, okay? We'll learn about what these graded potentials are. But it's going to get information either from a receptor or another cell itself, all right? And then its main goal, right, if this is the cell body and you've got these, we're called dendrites, these are these processes that come off. And then you got one big process, which is the axon, all right? Our main goal is to get a signal to the axon. Okay, somehow you can get a signal over here, but we've got to get it all the way over to the axon here, and that will start our what we call an action potential, our nerve signal. Okay, so this is what we're talking about. We want to conduct these little signals to the axon. Okay, we'll get there somehow. Another substance that you do need to know is called this chromatophilic substance, also known as Nissl bodies. Okay, these are just going to be, again, they're ribosomes. But we give them these names, you know, the gentleman that actually discovered them was Dr. Heisenfeld Nissl, all right? Actually, I don't know his first name. I just made that up. But anyways, I do know that his last name was Nissl, and he actually discovered, all right, the ribosomes in the neuron there. Okay? Dendrites. This is important that you know this, okay? Dendrites are unmyelinated, all right? Myelin is a fatty covering that we find on cells. It helps to speed up, does several things. It helps to speed up the action potential. Dendrites are not myelinated, okay? They come off of the cell body. They're smaller, shorter processes that come off the cell body, all right? Their main job is to receive, all right, input information from another neuron or something, or stimulus of some sort, and then they're going to transmit that information to the cell body, and then eventually hope to get it to the Axon Hillock, or the axon here. Okay, trust me, we'll get into all the actual physiology later on. All right, the axon. All right, this is the large process that comes off of the cell body. This is pretty much that cable line, that phone line that we see. It's harder to teach this stuff because everyone has a cell phone now. Hardly anybody has a landline anymore. All right. So the axon, right, is going to be where our signal is going to transmit from the cell body all the way down to the terminal end of the axon there. The very first part, you need to know this, is the axon hillock. All right. So here's your cell body. and then this triangular portion where the cell body starts to taper down into the axon, all right, that's this space right here, that's the axon hillock. This is very important. The axon hillock is where we start an action potential. You need to know that, okay? It's called the initial segment, all right? But the axon hillock is where we start an action potential. I've seen test questions on that. So when we move into the axon, okay, our cytoplasm in the axon is referred to as axoplasm, so we changed the name of that again, all right? And instead of calling it the plasma membrane, all right, in the axon, we call it the axolemma, okay? So I'm going to come back to this slide here, quick picture here. All right, so here's your cell body. Here are these smaller processes. These are the dendrites. Okay, notice there's no myelin on the dendrites. These blue things, that's supposed to be myelin. Cell body has no myelin, all right, and the dendrites don't have any myelin. All right, more on that later. That's going to come into play later on. Okay, so then the big process we see, all right, where it starts to taper down, that's the axon hillock here. All right, and this is where your action potential starts. Okay, and then the action potential is that nerve signal, and it runs down the axon. Now, some axons have what we call axon collaterals, and these are just branches that come off. Okay, you could have hundreds of axon collaterals coming off. All right, and then we get all the way down to this end, to the terminal end, all right, and we have these little bulbs here, and those are your synaptic knobs. And inside the synaptic knob, okay, you have these little bubbles. Okay, those are called synaptic vesicles, and they're filled with neurotransmitter. Okay, norepinephrine, epinephrine, serotonin. There's well over 100 different classes. All right, we'll talk about them. We'll talk a lot about acetylcholine. All right, but that's that neurotransmitter. All right, and these bubbles move to the end here, and then they release their chemicals into this cleft here, this gap. And those chemicals will stimulate something to occur at these channels here. Hopefully it'll have them to open up. And we'll get into that later on. All right. So those axon collaterals are those little branches that we see that come off the axon. Okay. And then when we get to the very end of the axon, okay, where the axon ends, we refer to that as the telodendra. And on the tips of those little telodendra, those little processes there, that's where we see the synaptic knobs. Sometimes they're called synaptic end knobs. Right? That's where we're going to find our neurotransmitter in there, okay? Because we have our synaptic vesicles sitting right inside. More on all that. I don't want you to think I'm just kind of skipping through this stuff, but I'm trying not to repeat myself. All right. So we do have some classifications for our neurons. And this is something that you definitely want to know. The one that we're going to be talking about the most, all right, is going to be the first one, the multipolar neuron. Okay, that's the most common type. So in a lot of our examples, we'll be referring to this, meaning we have all these, that's what you saw here in this picture. Okay, that's a multipolar. You got a bunch of dendrites, and then you've got one axon coming off. Okay. So that's the most common one. That's the one that I'll use the most in my examples here. Okay? So multipolar, many dendrites, one axon. Bipolar, all right? That's my best friend's mom, all right? No, that's really true. She is bipolar, but bipolar is where you're going to have, all right, one dendrite and one axon. All right, so you're going to see, okay, you have your cell body, and then you've got one process going one way and another process coming off the other way, all right? And that's a bipolar, okay? One is going to be the axon, the other one's going to be the dendrite. A unipolar, right, you've got a cell body that sits up here like this, and then you got one little process coming off, and then it goes this way, and it goes this way, okay? So one of these processes is what we're going to refer to as a peripheral process, and then the other one is going to be a central process. We see this type of neuron in our dorsal root ganglia. What's the dorsal root ganglia? This right here. Dorsal root ganglia is this structure right here. It's in close proximity, all right, to our spinal cord, okay? But our sensory nerves, all right, are a unipolar type of neuron, all right? More on that later. Oops, not there. And then the last one, okay, is referred to anaxonic. That's easy, all right? An meaning nothing, so there's no axons. So this type of cell just looks like it has a bunch of dendrites popping off of it. No axons. Okay? All right, that's the structural classification. The functional classification, to me, is a little bit more interesting, right? You heard me refer to these before, all right? Sensory neurons and motor neurons. You need to know, all right, their other names. When we're talking about sensory neurons, they're also referred to as afferent with an A neuron. That means that they are going to bring sensory information from your periphery, from your eyes, from your skin, all right, from your ears, whatever, and it's going to transmit that information to your central nervous system, okay? So you'll have receptors in somatic and visceral regions. Somatic is what you're consciously percepting right now, all right? You can feel how cold it is in this room. You can feel how hard the chair is that you're sitting at, all right? Visceral receptors, all right? You don't really know those sensations, how much stretch is going on in your small intestine wall, all right? How much or how little your blood vessels are vasoconstricting or vasodilating, all right, depending on what your blood pressure is, okay? So these types of neurons are unipolar, all right? That's the one I was telling you about, all right? They have the cell body, and then it's got one process going one way and another process going the other way, okay? Motor neurons, efferent. Easy way to remember that is when you affect something, okay? You're doing something, muscles do something, all right? They're effective, okay? So motor neurons are efferent neurons, okay? So they're going to transmit information that's going to come from your brain or your spinal cord, all right, out to the periphery, out to your effector organs, all right? And that includes somatic, which is, again, what you have voluntary conscious control over. So that would include your skeletal muscles, all right? And then you have your visceral effectors. That would be glands. That would be smooth muscle. That would be cardiac muscle. Those are going to be your visceral effectors. These types, motor neurons, are multipolar. That's the one that we saw before. And they'll have collaterals coming off the axons too. Don't be fooled, okay? And then the last type of neuron is the interneurons. These guys rock, all right? They make up the majority of your nervous tissue, believe it or not, okay? And what I love about interneurons is that they not only receive information, all right, but they can process it, okay? They can process it on their own, and then they can figure out how they're going to react to whatever that information is. So they integrate that information and then influence the effect of other neurons somewhere else, okay? So basically, these interneurons allow communication between the sensory neurons and the motor neurons. They can either shut down whatever the effect is, or they can actually activate something, all right? So we'll see these guys only here within the central nervous system, okay? And these also, I should say, excuse me, are multipolar. Okay, we'll talk more about them. I know it can be it's easy to be overwhelmed at this information that I'm throwing at you, but I can promise you, with the extra 2 days I believe that I have to teach this stuff, you're going to own this like a boss. Okay, so going on and talking about the classification of our neurons, when we're talking about the physiology, all right, the functional attributes of these neurons. All right, you've got your sensory neuron set up in the skin, all right, or if you want to go the visceral route, in the stomach wall, all right. There's an increase in the stomach wall stretch, but some sort of stimulus is going to affect, all right, that sensory receptor, that sensory nerve. It's going to send its information here to the spinal cord, okay. And what it'll do is it'll synapse onto this purple nerve here. That's an interneuron. So that interneuron then can either inhibit the motor neuron, which is this neuron here, or it can stimulate it. So it's going to receive that information, it processes that information, and then it can determine if it wants to stimulate this motor neuron to do something, either to a skeletal muscle or to smooth muscle, cardiac muscle, or gland. Okay? So keep in mind, if anything else, that sensory input goes from the periphery to the central nervous system, and motor input, or output I should say, is going to take information from the central nervous system and bring it to an effector organ, all right? Muscle or gland. All right, so for our glial cells, these are these other types of cells. This is like think of the neuron, all right, as the superstar, okay, like the diva. And this is the diva's entourage, okay? So these type of cells have no ability to transmit information. So therefore, they're non-excitable, okay? They don't transmit information. Their job is to support and protect neurons. That's what they do, okay? And so we see them in both the peripheral and the central nervous system, right? They're not going to be, you can have a neuron that can be as long as your leg or as long as your arm. Those cells become very, very long, all right? So these glial cells are much smaller, but they far outnumber the amount of neurons in your body, all right? And they make up almost, if not more than half, of the total volume of your nervous system, okay? Big difference between these cells is that neurons are amitotic. They can't divide, but these guys can, all right? And they can actually regenerate if they become damaged. Neurons have a difficult time. Central nervous system neurons have a hard time regenerating, okay? So their job, all right, is going to be to help provide nourishment, help remove waste. They protect the neurons, all right? So in some situations, if a neuron gets damaged to the point where it cannot repair itself, these glial cells will actually infiltrate the space that neuron once existed and kind of act as a space filler or a seat filler, okay? Huge part, and if we have time, we'll talk about it. But during that time when you are, when the nerves are developing inside of you as a baby and whatnot, and as you're developing, all right, embryologically, these microglial cells and these neuroglial cells help to guide, all right, the development of that neuron tissue there. It's really kind of cool. All right. Some of the glial cells that you definitely want to know, especially this one here, the astrocyte, okay? The astrocytes are one of the more popular ones. I've seen it asked on several questions on tests because of this concept right here. Has anyone ever heard of the blood-brain barrier, the triple B? Blood-brain barrier is necessary to prevent the passage of certain possible harmful or toxic substances into your brain, all right? But it will only allow certain things to get into your brain's blood supply, all right? I'll talk about it a little bit. First of all, astrocytes, okay? I'm going to show you a nice picture here. It's a star-shaped cell. It's this green fella. All right, so here you got your astrocyte. And it has these structures at the end of their processes. These are called perivascular feet, okay? So these perivascular feet attach onto the endothelium. Remember the endothelium? That's the actual epithelium that is going to make up, all right, your capillary. Because remember, capillary is only one cell layer thick, all right? It's simple squamous epithelium, okay? So these perivascular feet attach onto that endothelium, and they can manipulate, all right, the spaces in between the endothelium. So they can make your capillaries more leaky, or they can make them tighter. It's really kind of an awesome type of concept that they have, right? So that's why it's so effective in the blood-brain barrier here. And that's how your actual glial cells are able to monitor what's able to pass from your blood into your brain tissue there, okay? Already mentioned that. Okay. Ependymal cells. Ependymal cells we'll talk in a little bit more detail when we talk about cerebral spinal fluid. Has anyone ever heard of cerebral spinal fluid? That's basically the fluid that your brain and your spinal cord bathe in, okay? It's got a lot of different functions, helps to act as a shock absorber for the brain and spinal cord. What it really does is it actually makes your brain buoyant, so it's literally floating in your skull, all right? And it actually reduces, or I don't know how you want to, I don't want to say reduces the buoyancy, but it makes the brain 95% more buoyant, because if it didn't transmit that property onto the brain, if you didn't have this cerebral spinal fluid, your brain would pour out of your foramen magnum, literally, okay? So, ependymal cells, right, are going to make the cerebral spinal fluid. All right, they're part of this structure here called the choroid plexus. And the choroid plexus is just essentially a filtering mechanism that we find around some of the capillaries up in our skulls there, and it pretty much filters out some of your blood plasma, because cerebral spinal fluid has a very similar composition to that of your blood plasma. So the ependymal cells pretty much act as a filtering mechanism, so we get our cerebral spinal fluid, all right, floating around. And if we have time, we'll talk about where it goes and how it floats, or how it travels through your around your skull and your spinal cord there. All right, microglia. All right, these are the cells that are going to initiate an immune response. All right, they're very similar to a smaller version of a macrophage. All right, all their job is just to wander around, all right, the central nervous system. All right, and if they come across any pathogens, then they will actually destroy the pathogen. If there's any damage, any other glial cells that are damaged, they'll come in and remove any of those damaged glial cells or damaged tissues up there, okay? The oligodendrocytes, when we didn't get a chance to talk about the peripheral nervous system cell, but you have, give me a second here, myelin, all right? You have myelin. Myelin is a fatty sheath that covers these neurons, all right? So in the peripheral nervous system, the cell that produces myelin is called the Schwann cell, all right? But in the central nervous system, we have the oligodendrocyte. And what this cell does, and I'll show you a nice picture, is that it's going to have these little processes that it kind of extends out from the body, and it wraps itself around, all right, some of the neurons there. So here's your oligodendrocyte, and so it sends out this process, and it wraps, all right, that axon with a myelin sheath, so it can help to increase, all right, the actual speed of the nerve signal there. And then in the peripheral nervous system, all right, we have our Schwann cells. So you can either call Schwann cells or Neurilemmocytes, okay? But in this situation in the peripheral nervous system, the actual cell itself will literally wrap itself around the axon, where when we were in the central nervous system, the processes from the oligodendrocyte would wrap around, all right, the axon. But in the peripheral nervous system, the Schwann cell, the whole cell itself will wrap itself around the whole axon process there, okay? All right. All right, I'm going to stop here for that. And what we'll do is we'll do some labeling here.
It's the whole lecture test actually, that's what you said, right?
No, no, no. This, if any of this information is going to be on the third, it'll be on the third lecture test. Yeah. All right, let's just go in. We'll do some labeling, and then I'll let you guys get on home to take your test if you haven't taken it yet. All right. So again, this is chapter 12. Okay. All right. So, two possible answers you can utilize when describing the cell body for the neuron. Obviously, you can call it the cell body or you can call it the soma. All right. That's where you're going to find a lot of the organelles. All right. Obviously, the nucleus. Okay. Then you'll have the endoplasmic reticulum wandering around. Okay. And then, what page is this on? Anybody? 41. 41. And then you can see here, if we kind of zoom in a little bit, you can see some purple dots, all right, or red dots, and those are going to be your Nissl bodies. Okay, so obviously if they're attached onto the endoplasmic reticulum, that'll be a rough endoplasmic reticulum, those will be bound, but if you see some floating around, which maybe that's a free ribosome, okay, but again, the Nissl bodies are we'll see, I got a slide for that. Chromatophilic substance. All right, here's the nucleus. To me, in this model, it looks like one of those golf balls that you would play golf at night with. Has anyone ever played golf at night before? Take a little glow stick, you break it up, and you stick it inside the golf ball, and the golf ball glows. Yeah, you can play at night. I don't know why anybody would play at night, though.
Yes.
I know, exactly. And I would not play down here because there's crocodiles and snakes. All right, so there's the nucleus there. Going over here now to the axon, if I zoom in a little bit here, all right, this red structure that it's pointing to, that is a mitochondria. All right, not these blue things. Okay, these red structures, those are the mitochondria. What's a mitochondria do?
Powerhouse of the cell.
Powerhouse of the cell, making ATP. All right, here you see your Nissl bodies or your chromatophilic substance. Now you guys can't say Nissl bodies, you have to write out chromatophilic substance. I'm just kidding. I'm joking. All right. So those Nissl bodies, that's what that arrow is pointing to, or it should be pointing to. It should be pointing to the little red, but of course it's not. All right, but it is going to be pointing to the red. All right, so we don't see the full structure because it's cut off, but these white processes that are coming off, all right, those are the dendrites. And again, their main job is to receive information and transmit that information to the cell body. Okay? So that's a dendrite. All right, so for this slide, okay, the axon, all right, is this whole, imagine the axon as your cell phone charging cable, okay? So the axon is this white structure, okay? And then everything inside of it. So think of an axon like a cord, okay? Like your charging cord, okay? That's the axon, okay? You call your cell phone charging cord your cell phone charging cord. Well, we're going to call this cord-like structure the axon. We're dealing with the nerve. Now notice, watch what happens to the arrow. It doesn't move, okay? The axolemma is the actual white part that you see here. It does not include everything else inside. So it's just like your cell phone charger, okay? That white or black, whatever color it is, that rubbery part on the outside, okay? That would be the axolemma to your cell phone charging cable, okay? The axolemma, remember, is the plasma membrane, okay? That's what we call the plasma membrane in the axon. We call it the axolemma, okay?
So like with the last and say, what is this white substance or versus what is this?
It would be specific, and if it's not specific, in my grading, I usually allow, if someone wrote axon and it was marked wrong because they wanted axolemma, unless there's words there describing it, because it'll be pretty specific, then either answer would be acceptable, okay? But what we're seeing inside here, this is the axoplasm, all that in there. Okay, but the axon lemma is going to be the white covering. Okay, so we go down to the far end of our neuron. All right, your neuron eventually will have, all right, these little processes that are coming off. Okay, and those processes, these little bundles here, those are called telodendra. Okay, so these tan, all right, or whatever brownish-like structures that you're seeing, these are telodendra from another neuron. Okay, they came from another neuron, right, and they're actually attaching onto this neuron. So the telodendra is going to be that slender-like process. It does not include what I'm circling. That's something else. Okay, it only includes this part here. Right, not that. All right. That is going to be called the synaptic knob or synaptic endbulb. That's where the neurotransmitters are going to be located. So if we zoom in, if you look closely on this one, you can see here's the telodendra. All right, then you've got your synaptic knob here, or synaptic end knob, and these little white dots that you see in there, those are those synaptic vesicles. That has all the neurotransmitter, and eventually, all right, it's going to get released here onto the cell membrane here. Okay. So going to the next structure. All right, inside that box, that triangular-like structure, that's the axon hillock. That is where your action potential starts. It gets generated there. That's a test question. I can't guarantee it because it's a randomized test, but it's in there. It's in the test pool. I can guarantee you. All right, the axon hillock. You need to know that's where the action potential starts. All right. Moving further down into the axon region, all right, this arrow is pointing, so read the question. If it says name the cell, all right, then you're going to either call it a Schwann cell or a Neurolemmocyte. Okay? Now, if it were to say, well, it won't say it, so you don't have to worry about it. Okay? Schwann cell or Neurolemmocyte. All right, and then of course there's the nucleus of the Schwann cell.
Could you call that the other word for it?
Neurolemmocyte?
Could you call it Neurolemmocyte?
Oh yeah, of course. Yeah.
Really?
Yeah, if you want to. Oh yeah. All right. And then you look at this one here. Now again, it's pointing to the Schwann cell. So again, this is going to be a specific question. If it's asking you for, if it says name the cell, then name the cell. But if it doesn't say that, then you can say, all right, myelin or myelin sheath. Okay? Unless the question asks for something different. But if it's pointing right to this and it doesn't ask you a thing, then I will accept, all right, myelin sheath or Schwann cell, okay? But normally, all right, the question will ask you, name the cell, okay? All right, and then from this little indentation to the next indentation, all right, refer to that whole structure as an internode. All right, and then finally, this gray-like structure, all right, made up of areolar connective tissue, that's that loose connective tissue, That is the endoneurium. That's the innermost layer, okay, that covers each individual neuron.
Do the lines have any significance?
It's just showing you that's loose connective tissue. It's their best approach at it. Those are collagen fibers. That's it. Told you. It's not a lot so far. All right. So let's cut it for here for right now, because I know that some of you. What was the second to last one again? This one or this one? That one. Yep. Endoneurium. You guys have any questions for me? Both at home or here? You're very welcome. All right, so we'll stop.